Before we look at the structure of the replication complex, I'd like to draw your attention to a table from your textbook that defines the proteins involved in E. coli DNA replication. So those that are shown at the top and here are the enzymes, many of which we've talked about, that are involved in initiation of DNA replication, and Pol 1, which is required for the gap filling. But I want to focus right now on DNA Pol 3. This is a very complex enzyme with a large number of subunits. As you can see here, they have been given Greek letter designations. These are the genes that are known to encode them, and you will often find that the name of the gene is also used to name the protein, which makes it a little bit easier than sometimes than remembering the subunit names with the Greek letters. So DNA E is the actual polymerase activity itself, but I want to draw your attention to DNA Q, which is an enzyme activity associated with the holoenzyme, and it has a 3' to 5' exonuclease activity. So that means as polymerase is synthesizing 5' to 3', it also has an exonuclease that can remove nucleotides 3' to 5', which means it's going backwards. So this is used for proofreading. It means that once a base has been inserted, if it doesn't fit properly, if it's the wrong base, DNA polymerase itself, using DNA Q subunit, is able to edit and remove the incorrect base. Another subunit that's very important is DNA N, which encodes the beta subunit, and this is called the sliding clamp. And this clamp is very important for processivity. So processivity is how well an enzyme stays attached to its substrate in order to complete its job. So increased processivity of DNA polymerase means that it binds to the DNA and it stays there long enough to synthesize a fairly long piece of DNA before it detaches. So all enzymes have the ability both to attach and to detach. And the detaching is just as important as the attaching, because if it doesn't ever detach, then it can never be released to perform its function again on a different molecule. So more processive enzymes for replication or for transcription are enzymes that stay attached longer, which means that they are able to make a longer product than ones that are not as processive. Some of the other proteins are involved in binding single-stranded binding protein, or loading the clamp, or holding the whole C on, and I'm not going to spend much time on those. They're not particularly important for you to know, but understanding some of the key parts of it in terms of structure and function is more important. This is a very elegant figure showing the relationship among the different proteins at the replication fork. So in yellow we have the polymerase shown here and here, and these two protein molecules are tethered together through a complex that's called the clamp loader complex, that's shown in green here, here, and here. The clamp loader complex holds one subunit of the clamp called the beta 2 clamp, shown here in red, and it loads onto each copy of polymerase a dimer of the beta 2 clamp. So you see it here as a complete circle and here as a complete circle. The beta clamp is responsible for holding the polymerase molecules onto the DNA, and hence it's very important in the processivity of the enzyme, its ability to hold on and continue to replicate. The clamp loader complex also makes contact with helicase, which you recall is the molecule that is opening up the DNA at the replication fork, and it also contacts DNA G, or primase. So in fact, it appears that all of the major proteins at the replication fork are held to each other: primase, helicase, polymerase, and clamp, and they are held together through this complex, which has the clamp loader at the center of it. So to orient you in terms of the location of these proteins on the DNA, this molecule is the leading strand of DNA, and this molecule is the lagging strand of DNA. So how do we figure out 5' and 3' ends? Well, if this is the leading strand, then the newly synthesized piece, which is right here, shown in light blue, is being synthesized 5' to 3'. So this is 5', and this is 3'. This then is 3', and the pink then is 5'. So this is the template strand, and it goes 3' to 5'. The newly synthesized strand, shown in blue, is 5' to 3'. So the new DNA is being made here. On the lagging strand, the old strand is shown in blue, and the newly synthesized strand is shown in pink. Notice that these two molecules are in opposite orientation to each other. So they're the same molecule. The newly synthesized DNA is coming out of this end, kind of the top of the polymerase here, just as it is here. So again, this is the 3' end of the newly synthesized strand. It's in pink. The other end of the newly synthesized strand, because this is an Okazaki fragment, still has attached to it an RNA primer, which is shown in orange. So this is the Okazaki fragment. Here is the primer, RNA primer, made by primase. And here's the new piece of DNA made 5' to 3' by Pol 3. Also associated with the lagging strand is primase, which has just made a new RNA primer right here, and in between, single-stranded binding protein bound to the single-strand DNA, protecting it from nucleases. This figure is from your textbook. It's very similar to the one I showed before in the previous slide, although not quite as pretty. It's got the same general structure, but has a few more features that I want to talk about. So the numbers here, number 1, number 2, and number 3, these indicates the site on the template DNA where DNA G, primase, makes a new RNA primer for an Okazaki fragment. The template sequence that's recognized by DNA G to create that primer is generally, almost always, ACTG. And you note that I am telling you this CTG because we always label DNA or RNA in a 5' to 3' direction, so 5' CTG. 2 is a second CTG, and 3 is a third CTG. So the RNA is shown here as a dashed line in blue, and that's the RNA primer that was made by primase. The DNA N is the sliding clamp, and that's also called beta 2, as I said in the previous slide. Although it's not at all clear here, it's a monomer on the sliding clamp, and it's a dimer that completely circles the DNA on the DNA polymerase. And helicase and primase, as I said in the previous slide, are shown here, and they are associated with each other and are held in part of the complex through the sliding clamp assembly unit. So let's look at how these two molecules of DNA polymerase, while working together, can lead to DNA that's being synthesized in two different directions. So if we focus on these primers 1, 2, 3, and 4, let's see what happens during replication. So this is the Okazaki fragment that was previously made. In B, we are now looking on the lagging strand at the DNA polymerase, and it is making the second Okazaki fragment starting with primer 2. So this is the second primer, and this is the newly synthesized strand being made 5' to 3'. So the DNA is being made on the lagging strand right here and displacing SSB, single-stranded binding protein, moving the newly synthesized DNA in this direction. So what happens when this newly synthesized DNA runs out of space because it butts into the end of the first Okazaki fragment? So at this point, the DNA right here has looped out. So it's this looping out that then allows the DNA to continue to be made discontinuously on the lagging strand. And then as DNA polymerase reaches this Okazaki fragment, it releases that DNA, so it lets go, and then it reassembles onto the third Okazaki fragment shown right here. So now DNA polymerase on the lagging strand has dropped this DNA, and it has now picked up the DNA with the primer associated with it. So the template here is the CTG, and the primer, shown here in blue, then becomes the 3' end to start synthesis of the third Okazaki fragment. Then that will continue to synthesize with the DNA that's being made looping out until it comes to the end of the second Okazaki fragment. Then the fourth primer will bind to the DNA after it releases the DNA it's just made, and this continues in a stepwise fashion. In the meantime, there is free sliding clamp that stays with the DNA when it's released. So the clamp has to attach a new clamp each time the DNA polymerase on the lagging strand releases the DNA. That requires a new clamp to be associated, so that's the clamp loading complex. So this dropping of the polymerase from the DNA leaves the sliding clamp associated, at least with the DNA, which then allows DNA polymerase 1 to use that sliding clamp to attach to degrade the RNA that's associated with the Okazaki fragment and to add new nucleotides to it. Termination of replication requires sites called TER sites, TER A, TER B, and not shown here, but probably also another site in E. coli at least called TER C, and a protein called TUS. The TUS protein binds to the TER sites and it blocks DNA B helicase, which of course is part of the replication complex at the fork, so that blocks further replication. This works at the replication fork shown here and here. So as the clockwise moving fork moves through TER A, it slows and it can't get past TER B. So these act as one-way gates. And similarly, going counterclockwise, the second termination fork moves through TER B but cannot move through TER A. So when these two replication forks move past the TER A, TER B, they then meet each other at some point, and where they meet is where replication terminates, so that the whole chromosome has been replicated, and that ending of termination in the region between TER B and TER A is what's mediated by TUS.